Any questions about the end of the immunology chapter? No? Okay, let's roll. Respiratory system. You're working on that in lab, so lecture and lab are pretty close right now, which is kind of nice. We're going to talk first about the structural and then the functional organization of our respiratory system. The structural is in the blue on the left and the functional is in the yellow on the right. We divide the structural into our upper respiratory system and into our lower respiratory system, with the cutoff line being right there at the larynx. So everything of the larynx and below belongs to our lower respiratory system. So let's go ahead and fill in and put in some notes for structural upper respiratory tract. Is going to be the nose, nasal cavity, not seen in the upper diagram, but we should add this, paranasal sinuses, because sinus infection is part of an upper respiratory tract infection, and pharynx, basically your throat. Lower respiratory tract is going to be the larynx, trachea, and I'm going to put down the bronchial tree because it's shorter to do that. When I say bronchial tree, I'm talking about the main stem bronchus, or primary bronchi, secondary bronchi, tertiary bronchi, until they get so small that you then come down to the bronchioles. Difference between a bronchus and a bronchiole: one of them has cartilage and the other does not. So which one does not have cartilage? Bronchioles. And so what's keeping them open patent is actually the smooth muscle, which is going to increase in percentage of the wall as we go further on down the tree. So bronchial tree is all of those structures, and then we are going to also add respiratory zone. That's over here on the right-hand side, because that's going to comprise the respiratory bronchiole, the alveolar duct, and then the alveoli. One of the things that's important for a respiratory zone structure, there is always an alveolus attached to the structure. An alveolus is just a little sac. It's an air sac. It's the absolute smallest structure of our airways. Going up and taking a look at the conducting zone, that's everything that moves air. That's important about the conducting zone. It's what's moving air. In the respiratory zone, what we're having here is gas exchange. And when we go down into the notes portion of our lecture material, functional, two main divisions: conducting zone, all passages that conduct air to the exchange membrane. So remember, we are moving air. We are not exchanging gases in these areas. So again, if we just write some things down: nose, pharynx, larynx, trachea, and bronchial tree. The very last structure of the conducting zone is called a terminal bronchiole. A terminal bronchiole is the smallest bronchiole that's conducting, where no gas exchange occurs, and then gives rise to a respiratory bronchiole. And then alveolar ducts, and alveoli. Again, this is gas exchange. is the hallmark of respiratory zone. Conducting zone, no gas exchange, just the actual movement of the gases through these structures that were listed. Looking at our conducting zone structures, let's see what they're there for. Our nose is actually designed to help moisten, which is humidify, warm, and filter the air that's coming in. It also serves as a resonating chamber for sound production and is our area where our olfactory receptors are located up high. Structurally, when we take a look, air comes in through the external nares or nostrils, then enters into the nasal cavity, which is divided by the nasal septum, which is bony and cartilaginous. What type of cartilage is it? Hyaline. That's your go-to cartilage, right? Hyaline is almost like a knee-jerk response. In the roof of the nasal cavity, that's where the ethmoid bone is. The floor of the nasal cavity is called the hard palate. Remember, the bulk of the hard palate is our maxillary bone, and then those little palatine bones are on the posterior aspect of that hard palate. The mucosa that lines the hard palate is ciliated columnar epithelium, and it has lots of goblet cells, which are going to secrete mucus and trap particles. So that's part of that filtration, getting the stuff out of the air before it goes down into the smaller airways. Which way does the mucus move in our nasal cavity? Yeah, backwards, right? The only time it moves forward is when we have a lot of inflammation, right? And it's overwhelmed. And so, or when we cry, kind of things. So the only reason it comes forward is because the system is overwhelmed, and so it comes out both ends, but goes backwards into the pharynx itself. The concha are 3 on each side, come off laterally and project into the nasal cavity medially. We have a superior nasal concha, a middle nasal concha, and inferior nasal concha. And these will increase the surface area of our nasal cavity and increase turbulence. Increasing the turbulence of the air helps to give us a better sense of olfaction, but also increasing the turbulence allows for vortexing and more humidification, as well as warming and filtering of particles. Pharynx, connection of the mouth to the larynx and esophagus. There's a little bit more to it than that. We have 3 subdivisions of the pharynx. We have the nasopharynx, the oropharynx, and the laryngopharynx. And they're associated with the nasal cavity anteriorly, the oral cavity anteriorly, and then the larynx also anteriorly. When we take a look at the nasopharynx in our cartoon drawing here, the major structures that I want you to keep in mind that are part of it are the pharyngeal tonsils, as well as our auditory tube is actually opening into that. Our nasopharynx is continuous with the nasal cavity, which is anterior to it. And again, pharyngeal tonsils and auditory tube. And you remember the auditory tube has a couple of names, right? Pharyngotympanic tube, eustachian tube, all of those are the same as the auditory tube. Oropharynx, continuous with the oral cavity. Again, that's an anterior, the oral cavity is. And in here we have the palatine and lingual tonsils. And then laryngopharynx, associated with the top of the larynx. Air goes into trachea, and food goes into the esophagus. And so at the laryngopharynx, this is the area where decision is made. As to whether we're going to allow it to go into the trachea or we're going to allow it to go into the esophagus. And one of them has what we call the right-of-way, has priority treatment. And sometimes it's not exactly what you think it is. Which one has priority treatment? Food or air? What can you, I want you to think about this, and that's why you have to actually think about this. When you breathe, does it matter if it goes into your stomach, air? But when you swallow, does it matter if it goes into your airway? Yes. Who has priority now? Food. Food control. Food is the determining factor. If you have food, you stop breathing when you swallow, correct? When you're swallowing food, you will actually temporarily not do any sort of breathing. Okay, so that's an important aspect. So food has the right of way. What happens when food gets in your airway? You start coughing automatically, and so you actually are trying to expel that at that time because it's not supposed to be there. We'll talk a little bit more about that cough reflex in just a moment. Let's go ahead and take a look at the larynx, which is what you guys are dissecting out on the cat. When you view the larynx, you are typically viewing the laryngeal area from an anterior aspect, correct? You can't view it from a posterior aspect unless you dissect it out, because what's behind the larynx? The esophagus is behind the larynx. So let's take a look at the structure of the larynx and realize that we have a lot of cartilage here, but there are only three cartilage structures that are actually very obvious and easy to find. The other cartilaginous structures are really, really small and difficult to find, especially in your cat. It's almost impossible to find in your cat. We call the larynx the voice box because that's where vocalization actually occurs. It is indeed our switch box for air and food as well as sound production. We have 9 cartilages. We have 9 cartilages that actually form its structure, and 3 of them are large. And of the large structures, we have the thyroid cartilage, which is singular as a large structure, right here, pointed out to it, this guy. And then we have the cricoid cartilage. And the cricoid cartilage is not very large anteriorly, but take a look at it posteriorly. Posteriorly, it's much larger than it is anteriorly, and take a look at what happens to our thyroid cartilage posteriorly. It is incomplete. It stops. Not so in your cat. In your cat, it actually is continuous posteriorly, but our thyroid cartilage actually is discontinuous on its posterior side. What kind of cartilage material is this thyroid and cricoid cartilage? Hyaline cartilage. So then we also have the epiglottis as part of the obvious cartilaginous structure. And what kind of cartilage is the epiglottis? Elastic. Here, you guys remember. Yeah, there's only 2 E cartilages in our body, right? Ear and epiglottis, which is really, really nice and easy to remember that. So that is indeed, it says series of 9 cartilages. So all the other cartilages are hyaline. It's just that the epiglottis itself is the only elastic cartilage. The cuneiform, the corniculate, and the arytenoid cartilages are really, really small and only can be viewed from the posterior aspect. And the arytenoids are the ones that are actually more or less attached to our vocal cords or ligaments. Notes here, thyroid cartilage contains the laryngeal prominence, which is also often referred to as the Adam's apple. The reason we like to actually discuss that is because if for some reason somebody has airway blockage superiorly, if you want to actually go ahead and create an artificial airway for them, you want to be able to find the laryngeal prominence of the thyroid cartilage. And then you'll go down below that, and the little divot that you'll encounter is this area called the cricothyroid ligament, and that's where you can pierce it and make a hole. You know, do that MacGyver stuff. Who doesn't know who MacGyver is? Are you willing to raise your hand? So MacGyver is that person that has this bag full of tricks because of knowledge and simplicity. And because of that knowledge and simplicity, knows that if somebody has an airway obstruction superiorly, you can create an artificial airway by actually piercing this because you have no structures in front of it that will cause you problems. That's why knowing the laryngeal prominence or the Adam's apple is very important, because truly you go down, you find that little divot, you stay in the midline, you can take a little knife and you can cut through all that stuff and take a, what is it, they use usually a pen, the pen container, and you put that in there, but you know, it's still got too little of a hole on one side. You're going to have to somehow break it in order to make that straw a little bit thicker in its diameter for it to work. But it suffices as a temporary airway until paramedics or emergency personnel arrive. The epiglottis is an interesting structure. It covers, it's called epiglottis, you know, like skin is the outer aspect. So epiglottis covers the glottic opening. When you swallow. The glottic opening is the passageway down into the rest of the larynx and the tracheal area. And so what is interesting is you guys can find your larynx. Go ahead and find your larynx. It's a little uncomfortable sometimes, but go ahead and, you know, just be gentle. You know, it's your own self. You can be gentle. So go ahead and swallow and see if it moves. Does the larynx move when you swallow? It should move. And which way? Down or up? It moves up. And that's important. So the larynx is shifted superiorly when you swallow. What that does is that upward movement causes the epiglottis to literally tip over the glottic opening. And that's an important aspect of swallowing, and that's your epiglottis is very important in that maneuver. So when you swallow, typically you're swallowing spit, water, food, things along that line. And so our little teeter-totter is going to tip over and it's going to cover that glottic opening during swallowing because we elevated it and caused the top to cover it. And therefore the food is deflected from going into our larynx and goes into the esophagus. So larynx pulled superiorly when swallowing, and epiglottis tips over the glottic opening, causing food or whatever that you're trying to swallow to be diverted into esophagus. If it doesn't work effectively, we will start a cough reflex, correct? If fails, this causes cough reflex. Is the cough reflex functional when you're unconscious? No. And that is one reason why when somebody's unconscious, they typically want you to turn them onto their side. So only works if conscious. Our vocal ligaments are elastic connective tissue on either side of the larynx. We commonly call them the vocal cords. We also have false vocal cords, which are not elastic CT and located superior to the true. False vocal cords have no role in sound production. And in our picture up above, these guys are the ones that most people will call our vocal cords, and those are the false vocal cords. Those are the first things that you see. In order to find the true vocal cords, you have to look internal in order to find them. Moving on down into now the trachea, and then we'll get into the bronchial tree. Trachea is referred to as your windpipe. It has several layers. Going from the luminal side out, we have the mucosa, then the submucosa, cartilaginous rings, and then adventitia. The mucosa is ciliated pseudostratified columnar epithelium with goblet cells. And now that we are in the trachea below the larynx, I want to ask you, which way does the cilia beat here? Which way with the cilia? Superior or inferior? It beats superiorly, because if it beat it inferiorly, remember the cilia is going to be moving things down if it beats inferiorly. Well, what's down there? Lungs. And do we want to send a bunch of crud on down into the lung tissue itself? We do not. What it does is it actually, our cilia beats superiorly. The nose beats posteriorly, the trachea beats superiorly, and it will take all that yucky that it's trapping, because that's what the mucus is for, and it will then put it into the esophagus. And so you'll swallow that mucus, which you do on a regular basis. You don't realize it. You think you're just swallowing saliva, but you're swallowing also mucus that is coming up from the lungs on a regular basis. It's not a big deal, because it goes into the esophagus and the stomach and the digestive tract, and if there's anything there that's got any nutritious value, you'll be able to harness it. But if it's mostly debris, it will be sent out with the rest of the debris. So it's actually a system that's remarkably efficient. The submucosa contains seromucous glands, which also produces most of the bulk of the mucus that we actually find there. One of the things I do want to point out about mucosa, remember it's epithelium plus a little layer of connective tissue that we call lamina propria. Tracheal cartilages, they are C-shaped rings. They're not completely circumferential. They are incomplete posteriorly. So C-shaped rings with the incomplete connection posteriorly, made out of hyaline cartilage. And we're going to draw this in just a moment. On the outermost aspect is adventitia, which is a tough connective tissue layer. So this is different. Adventitia is a thick CT layer instead of a serosa, which is just a thin, simple squamous epithelial layer with a little CT holding it on. Your tracheal cartilages, I'm going to do it kind of like this, are C-shaped. And so the incomplete area or connection is posterior. And I'm going to draw what's right behind, and that's our esophagus. And the esophagus is typically collapsed. That's one of the things that you guys should notice with your cat. Your cat's esophagus should be pretty darn flat, correct? It's very flat. At first, you don't even know it's an esophagus until you start messing with it. So the esophagus is a muscular tube and is ordinarily collapsed. It doesn't have an open or patent lumen unless There's food in there, something that you're swallowing. Now, your tracheal cartilages have a muscle right back here. It's called the trachealis muscle. What type of muscle do you suppose this is? It's a smooth muscle. So we have this trachealis muscle that's a smooth muscle that is typically somewhat contracted. And this helps to keep the two C cartilage where they are and keep the lumen of the trachea patent. However, when you swallow food, we just already talked about just a moment ago, what does your breathing do when you swallow food? It stops. There is a very good reason for this besides this little right-of-way thing, because now this guy back here is bulging into this area with this bolus of food in the middle of it. Well, it's literally bulging into the lumen. Ugh. Of course, everything goes away. It's bulging into the lumen here now. The trachealis muscle is doing this. So what did that muscle do? Did it contract or did it relax to allow food to relax? And what's going to do, it's going to relax in front of the bolus. You guys follow me with this? So before the food goes down, the trachealis muscle, which was contracted, will suddenly now relax. And this bolus of food will be able to easily move on down as this occurs. After the bolus of food moves down, what does the trachealis muscle then do? It contracts and then reestablishes the patency of the lumen of the trachea. So this is another really kind of unique and very effective way to actually ensure that you're supposed to stop breathing whenever you swallow food. And so then once the swallowing is done, then the trachealis muscle contracts, and then you actually have a patent airway again. So trachealis muscle, and then also the cartilaginous rings, or cartilage, is connected to each other superiorly or inferiorly by elastic CT. So as you take a look at the trachea, you can easily see the cartilaginous C-shaped rings on the trachea, but there's also a connection between each tracheal cartilage, and that material is actually elastic connective tissue. Going on down now into the bronchial tree, so bronchi and subdivisions. So we have a right and left primary bronchus for each one, and there's only one right and only one left, branching off at the trachea in the area of your sternal angle. So find your sternal angle on your chest, that area where you feel along your sternum and there's a protruding bump there. That's your sternal angle. Superior to that is the manubrium, inferior to that is the body of the sternum, and this is where our trachea ends and our primary bronchi begin. So it's kind of nice to actually know there's a simple landmark that you can actually figure out for that. So our primary bronchus is the first branch, then the second branch is called secondary bronchi, then after that's tertiary bronchi, quaternary bronchi, and as long as there's cartilage in the airway, it's a bronchus. As long as there's cartilage in the wall of the airway, it's considered a bronchus. When cartilage is no longer present, it is then called a bronchiole. It's important to note that our right main bronchus is more vertical than the left. And this means if you were to accidentally aspirate a foreign body, it most likely will lodge into the right main bronchus. Going up to the colored bronchial tree, of course, here's our trachea. Here is our primary or main bronchi. Then a branch. The next branch is the secondary or lobar bronchi. So I want you to know also secondary is the same thing as a lobar bronchus. And then off of the secondary, we will end up having then our tertiary, which is also our segmental bronchi. So primary is main, secondary is lobar, and tertiary is segmental bronchi. And that's typically when somebody has pneumonia, they'll say they have lobar pneumonia, which means that whole lobe of the lung actually is infected and has purulent material in it. The smaller bronchi, remember, still have cartilage, and that's what we're going to take a look at when we scroll on down here to changes in the wall. So again, our tracheal area here, elastic CT connecting the cartilaginous rings that are incomplete. However, once we get to our main bronchus, the cartilaginous rings are circumferential or complete. We still have elastic CT in between, but then as we then go down into the lobar area, take a look, we're getting now to be cartilaginous plates, and we're getting thinner and thinner as far as the cartilaginous material as we get smaller and smaller. Until finally, because here it is, smaller bronchi, very little cartilage, and then once we get into the bronchiole, there is no cartilage presence in the wall of that airway structure. So a bronchiole does not have any cartilage present. And that's part of the notes here. Cartilaginous rings are replaced with plates of cartilage as we go into the smaller airways. And when I say smaller airways, it means for sure that they have a smaller lumen or diameter of the lumen. The other thing too about the epithelium, we were columnar, pseudostratified ciliated columnar epithelium, and as we go into the smaller lumens, those tall cells are not going to be very useful, so we need to get shorter and squatter, and indeed the epithelium will become cuboidal. We're going to lose our cilia as well as our goblet cells as we get into the smaller and smaller areas. As we get into the smaller lumens, one of the other reasons that we lose those goblet cells is they produce that mucus, and we shouldn't need the mucus to trap things anymore as we're in the very, very small airways. All of the particles should have been taken out in the larger airways. When we take a look at the smooth muscle, that's part of the airway structure itself, we see percentage-wise. It increases percentage-wise as we get into the smaller and smaller tubes. And this is, smooth muscle is only structure keeping the airway patent or open. And the picture up above just shows bronchoconstriction, which is not something that's terribly desired in our smaller airways, and then also bronchodilation, a nice open lumen there. Bronchoconstriction is typically something that we see with people who have asthma. All of that was conducting zone, so no gas exchange in all of the structures that we were just talking about. Now we're getting into what's called the respiratory zone, and this is where gas exchange is actually going to occur. The gas exchange only occurs in that structure which is an actual alveolus. So when we talk about a respiratory bronchiole, right down here in the cartoon, you can see the little layer of smooth muscle as part of the respiratory bronchial wall, but there's an alveolus that's attached to it. And up here in our histology, we see the respiratory bronchial wall kind of up in here, parts of it up in here, which is thicker. And this guy right here is an alveolus, that little sac there. And so that's why it's called a respiratory bronchiole, because there is an area where gas exchange can occur. Alveolar ducts, they look like, as you're looking at lung tissue, it looks like there is a channel there, and so therefore it's called an alveolar duct. But then all these little areas that look like little sac-like structures are definitely all alveoli. And so they're not that difficult to identify. You get a little bit confused at times in trying to figure out what a respiratory bronchiole and an alveolar duct is. Remember, with a bronchiole, there is some smooth muscle present. So there is some more substance with a bronchiole. And an alveolar duct, there is actually no smooth muscle at all present in that structure. When you take a look at the cartoon of the getting deeper into the lung tissue where all these alveoli are located, down here at this area, take a look at how the capillaries act like a network or a hairnet over a balloon. And so the capillaries are very small in comparison to the alveolus itself. And the alveolus is literally a sac of air, and this is the area where we call the respiratory membrane, where gas exchange actually occurs. So this respiratory membrane is the wall of the alveolus, which is simple squamous epithelium. We'll also have basement membrane, and we'll have the wall of a nearby capillary, which is also simple squamous epithelium, which we also call endothelium. So we have two very flat cells with a basement membrane in between, and that is thin enough for us to actually allow gas exchange to actually occur. Let's take a look at some of the cartoon, further cartoon demonstrations of the respiratory membrane. Nice big sac here, alveolus. There are little connections between one alveolus and another. They're typically not all walled off all by themselves. And you can see this is meant to be a type 1 alveolar cell, nice big flat simple squamous epithelial cell. And then a few other cells will be inside the alveolus, these guys that have these protrusions. Those are type 2 cells. They're called cuboidal cells, and they are responsible for secreting a material called surfactant. Another cell type that will also be found wandering around in our alveoli, even at this very small structure, are alveolar macrophages, and they have an interesting little name for them. They're called dust cells. And so they're picking up whatever particles that actually made it down into these very, very small structures. The capillary is right there. Blood is flowing close to air so that we can actually have efficient gas exchange. A little bit of a description of the lungs and the pleural coverings. Pleura is also Latin for lungs. Each lung is suspended in its own pleural cavity. So we have a right lung and lung field and a left lung and lung field. Of the left lung, we only have 2 lobes, and the left lung, dimension-wise, is actually smaller than the right lung and has a notch for the heart. Each lung is divided into lobes, 2 on the left, 3 on the right, and this is by connective tissue, and each lobe itself is divided into smaller segments called bronchopulmonary segments via connective tissue. Each bronchopulmonary segment will be serviced by its own artery and vein. And so you'll find an airway, and on one side of the airway, this bronchopulmonary segment or bronchus, will be a vein on one side, and on the other side of that airway will be an artery. There's no gas exchange occurring at these larger structures, but the vein is bringing oxygen-rich blood back to the heart, and the artery is taking oxygen-poor blood into the deeper areas of the lung, to the alveolar areas and the capillaries, so that we can have gas exchange occur. Pleura, we have parietal and visceral layers. Parietal layer is on what surface? On the rib cage, so it's on the inside of the ribs, correct? And then the visceral is on the surface of the lungs. In between is the pleural cavity, and in the pleural cavity is some pleural fluid. And what color do you think the pleural fluid is? It's clear. Now, each lung has its own cavity, so if one lung is damaged, that means that the other lung is still functional. And that's why we have two kidneys too. If we have one that gets damaged, the other one still functions, and we're able to survive and live. And so we have a right lung field and cavity, and we have left lung field and cavity, and they're not connected. So whatever happens to the right is going to be only experienced by the right, and same for the left, unless of course it's some sort of ascending cancerous or pulmonary infection that's going up the bronchial tree backwards and then can pour into the other side. Pulmonary ventilation. Ventilation is a movement of air pattern. So pulmonary ventilation. When somebody's on a ventilator, is there gas exchange? Is the ventilator exchanging gas, or what is the ventilator doing? It's moving air. So pulmonary ventilation is air movement. Pulmonary respiration is gas exchange. So when we use respiration, we are talking about gas exchange at the cellular level. Inspiration, air is flowing into the lungs, also inhalation. So you inhale. Expiration, gases are leaving the lungs, so you are exhaling. And inhalation is an active process, and exhalation is a passive process for the most part. Let's talk about pressures in the thoracic cavity relative to atmospheric pressure, which is atmospheric pressure is 760 millimeters of mercury. We have two terms here, intrapulmonary pressure, so that's pressure within the alveoli or within the lungs itself. It is sometimes also referred to as intra-alveolar pressure. So intrapulmonary pressure, the pressure of the air in the lungs. Intrapleural pressure is the pressure in the pleural cavity and is almost always less than intrapulmonary pressure. And so this almost always is 4 millimeters of mercury less. Let's take a look at this story down here with this thoracic pressure relationship diagram or picture. Atmospheric pressure is all around us, correct? We are suspended in atmospheric pressure. What's the pressure inside your nose? Atmospheric pressure. What's the pressure inside your pharynx? Atmospheric pressure. What's the pressure inside your trachea? Atmospheric pressure. As long as there's nothing blocking it, right? Aren't we continuous with the atmosphere in all of these conducting zone structures? You guys following me with this? If you are a conducting zone structure and you are patent, your pressure inside you is the same as the pressure outside the world around you. And so that's what we have to understand here. This says 760 right here. So all of that is connected. So therefore the pressure there is always 760 millimeters of mercury unless we're doing something. So this is basically just right now at rest. So in our pleural cavity, the pressure is 756 millimeters of mercury. And so the pressure is greater inside the lungs than it is outside the lungs inside the pleural cavity. Where does pressure want to go? Does pressure want to go from low to high or high to low? High to low. And so what this is going to effectively do, this is going to effectively try and push, isn't it? Isn't the pressure that's inside the lungs going to try and push outward to the area that's lower? Correct? So this is one of the very important reasons why our lungs stay filling up the cavity. The pressure in the cavity is a little bit lower than the pressure in the lungs. So if the pressure in the lungs is higher, it wants to expand outwards into the cavity. You guys follow me with this? And so that's part of what helps to keep our lungs inflated. There are other things involved in this, but this is an important thing to realize as we take a look at these little diagrams here. Here's a piston, and it's showing you that you have a volume, and you have certain molecules inside that volume on the other side of the piston. And when you pull the piston up, which increases the volume of that space, you end up creating a lower pressure in there. When you push the piston down and decrease the volume, you have a tendency to increase the pressure. Correct? You guys follow that? So there's molecules in there. When you make, that's why it was important for you guys to remember this also from our cardiovascular chapter. When we inhale, our diaphragm goes down, increasing the volume of our thoracic cavity and decreasing the pressure. When we exhale, which is actually a passive process, we just relax, the diaphragm goes up, the volume goes down, the pressure goes up. And where will air move from? Air moves from a high pressure to a low pressure, correct? And so when we decrease the volume, we increase the pressure and the air moves out. And our diaphragm relaxing is a passive process. So when we inhale, our diaphragm contracts, increases the volume, decreases the pressure, and so air is pulled in. You guys follow me? So I'll commonly say air is sucked in. We literally create a negative, when you create a negative pressure, that's like suction to me. You guys with me on that one? And so we literally suck air in, and then we passively push air out, but passively push it out because of the relaxation of the diaphragm. And that's what these little charts are meant to actually show you, is that it's a volume-pressure relationship. Volume goes up, pressure goes down. Volume goes down, pressure goes up. It's an inverse relationship. We'll stop there. I'll bring the little diaphragm guy next time, and moving ahead nicely.